Howdy. Howdy is the traditional greeting here at Texas A&M University. And my name is Dr. Bill Klemm, and I'm a brain researcher in the College of Veterinary Medicine and Biomedical Sciences. Before we begin our presentation, I'd like to ask you a couple of questions. First of all, have any of you ever been sick enough to require pharmaceuticals to treat your illness? Or maybe you remember your vaccinations. Maybe you even get them every year for preventing flu. Have you ever asked yourself, where does this stuff come from? Does it really work? Is it safe? Well, the purpose of our presentation here today is to answer some of these questions and tell you about the process by which we determine whether or not pharmaceuticals and medical devices are safe and also effective. So let us get on with the presentation. And I want to remind you that during this presentation you will see a couple of interviews from Texas A&M scientists who participate in both preclinical trials and clinical trials as part of their research efforts. I think you'll find this very interesting.
Welcome to our virtual scientist visit to your classroom. We want to explain today how basic research turns into medicines and treatments. You notice our symbols here in this slide show the symbol for veterinary medicine and the symbol for human medicine. The symbols overlap to indicate that there really is only one medicine. And much of the basic research used in creating medicines and treatments is done on animals. One thing you may not realize about basic research is that it takes a long time for discoveries to become a pill or a medical device. Moreover, the odds of success are not good. In the example here, there was a study done on discoveries between 1979 and 1983, and during that period 101 discoveries were reported that could lead to a medical treatment. Yet only 5 of these received a license for clinical use, and of the 5, only 1 was a popular treatment. Even when an idea is successfully translated into a medicine or a medical device, it still takes a long time. As you can see here, for instance, the range is 14 to 44 years. 44 years! And the median is 24 years. So you can see that a company has a considerable investment in producing a medicine or a medical device. Next, I want to illustrate the process by which discoveries are translated. The first thing they have to do is identify a target. And by that I mean that for medical devices and appliances, for instance, you have to identify what the objective of the treatment is going to be. For instance, you might want to make a more effective or longer lasting artificial joints, or develop an artificial heart. So those kinds of things would be the target. For pharmaceuticals or drugs, you usually identify a biochemical reaction that controls the body function that needs the treatment. For example, insulin is known to regulate blood sugar, or another compound was discovered to control appetite, a compound called leptin. Well, these chemicals, biochemicals, are targets that for which you can develop drugs to manipulate their function. Here's an example using hydrogen sulfide. Yes, and that's the gas that you get from smelly eggs. And you would not think that there's some medical advantage of this gas. But actually the human body makes hydrogen sulfide too, and it turns out it's a signaling molecule that relaxes blood vessels. Well, if you have a target chemical like hydrogen sulfide that relaxes blood vessels, you also have a target for drugs that might lower blood pressure. by manipulating the level of hydrogen sulfide, you could affect blood pressure. Now, once you have a target for developing a new blood pressure medicine, the strategy might involve, for instance, developing a chemical that activates the enzymes that make hydrogen sulfide. Or you could make a drug that stimulates the formation of the body chemical that naturally activates the enzymes for hydrogen sulfide. And one of these targets is calcium calmodulin, a protein. And by manipulating the calcium calmodulin, you can control how much hydrogen sulfide is made. The development process itself obviously requires that you understand the science. And then, of course, you can develop a prospective treatment for the target that you've identified, that you presumably understand the science about. Next thing you have to do is develop an assay system. In other words, how are you going to measure the effect of the treatment? And then finally, you have to test it in a suitable animal model. And this is the stage at which you begin preclinical trials, almost always in animals. And if the animal trials indicate that your treatment is effective and safe, then you can begin a formal clinical trial process in humans. And eventually, after all the data are collected, you get approval from the Food and Drug Administration to market the product. At the College of Veterinary Medicine at Texas A&M, we have scientists who do clinical trials and preclinical trials in animals. And next in our virtual visit, we're going to show you some of their labs. I'd like to introduce you to Dr. Heather Wilson, who's a small animal clinician here at the College of Veterinary Medicine, and she specializes in cancer of dogs and cats. And we're in what you might call the war room, where they have records of the different cases and what their status is. And so maybe you'd like to tell us a little bit about what this room, what goes on in this room, and then we can talk about other things.
Sure. So here's where we actually take in all of our patients, and we do a lot of our treatments, draw blood and get samples. And so initially we have a board here that's set up for clinical trials. And all of these patients are enrolled in clinical trials. We're collecting samples, keeping track of them, administering medications and things at appropriate dates. And then this board is our regular patient board. And some of these dogs are also on clinical trials and in both cases, but most of the time these are the patients we're going to see each day, their names, when their appointments are, what their problem is, and then what the plan of attack is for the day as far as what treatments or diagnostics they're going to get for their workup. And so as the day goes on, we check the board off and then send them home.
I'm sure they're all nice little tumors.
We're in Dr. Wilson's lab right now, and we wanted to explain to you a little bit about how she does the clinical trial work here in the small animal clinic. And you do, as I understand it, clinical trials for drug development for dogs and cats, right?
Yes, sir.
Among other things you do, which we'll talk about in a minute. Could you explain a little bit about some of the equipment you have there in the lab and how you use it in your clinical trial tests?
Sure. So the clinical trials that we currently have going on involve many facets of veterinary oncology, and one of them is simply looking at the bacteria in the gut and comparing it to before and after chemo, because chemotherapy can cause diarrhea, and we want to find out why exactly that is. And so that's a simple matter of collecting fecal samples and looking at the DNA of the different bacteria that are in those samples before and after. And that's done using very simple tests and some of the equipment here, which I'll explain in a minute. Additionally, we are doing tests for dogs with lymphoma, where we're actually taking lymphocytes or white blood cells, kind of the army of the body that's used to protect the body from infection and other things. We're trying to take those cells and make it so that they will actually attack the dog's tumor. And so we're going to draw blood and take those cells out of the blood and then introduce them to the tumor in a way that would make them recognize it as something bad. and then we can put it back into the dog through an infusion, like an IV infusion, and then hopefully those cells will remember what we've taught them and attack the tumor cells in the dog's body just like they would if it were a bacterial infection. And that's one that is currently going on now. And then in addition to that, we also have another clinical trial for dogs with melanoma. And melanoma is a tumor of pigmented cells, the ones that make you tan when you go out into the sun. And those particular tumor cells are pretty aggressive, but a lot of times we can get the body to recognize them also as something that's bad, like a bacteria. And so we're going to give these guys drugs that are just like chemicals in the body that stimulate the immune system. And we're going to attach those little chemicals to antibodies that are specific for that type of tumor, so that when we give it to them, the antibodies will kind of taxi the little chemicals straight to the tumor, so that the immune system can recognize those chemicals and know that's where they're supposed to attack. And so those are the three kind of trials we currently have going on in oncology. How we would use this equipment associated with that depends on the type of tumor. This right here is a thermocycler. It's a PCR machine, or polymerase chain reaction. What you do with this machine is you actually look for DNA of a specific gene. And so if there's a gene that you think might be mutated in cancer, you can use this machine to try to find it specifically in a sample. And so for tests like the fecal test that we're talking about, we could use this machine to identify DNA. Also, for any other type of tumor, if you want to know if a specific mutation is associated with it, you can use this, and this can find that particular type of DNA in a very, very, very small amount of tissue by amplifying it thousands and thousands of times, so that even the smallest bit of DNA can be found. This piece of equipment here is a simple centrifuge, but it's refrigerated and lets us centrifuge cells and samples at a temperature that keeps them from breaking down, and is useful for things like PCR and other tests. Here is an incubator. Now we use incubators for cell culture, and when we do cell culture techniques, essentially we keep them on little racks in here, and it's humidified. There's a tray of water down here to keep the humidity up, and it's kept at body temperature, and it's kept at a carbon dioxide level that is similar to that, so that these cells have an environment that's happy for them, much like the body, that allows them to grow and proliferate, so that we can take cancer cells, and just the cancer cells, and study them individually without having to use any live animals to study cancer. This here is a sterile hood, and what we do in this hood is we do cell culture techniques. So when we have the cells and we pull them out of the incubator, if we want to collect them, or if we want to put them in a petri dish, or do anything special like count them, we do it all under here wearing gloves, and you sit in under here. There's a UV light to help sterilize it that we can turn on here. The UV light will sterilize this area, and then when you work with the cells, you just turn the regular light and the fan on, and that fan keeps the air going this way. Keeps the air going this way, so that no air from the outside can blow in and contaminate your cells, so that everything stays clean under here. And so we work this way, usually with sterile pipettes and things to work under with our cells. This here is a liquid nitrogen tank. Liquid nitrogen tanks are used for long-term storage of cells and cell culture. And in this tank, you essentially have several different cells cell lines that we keep in here frozen, and they can remain frozen for years this way, so that when we need them, we can pull them out, and then if we don't need them, they can stay in here and alive until we pull them out. Or we have a box here with about 15 different cell lines, all from canine, feline, and human tumor cells that were developed from real tumors that happened in real animals or real people. And so that's how we keep them, so that we can use them. And then here is just a simple water bath. And these are the ways we heat these cells up. So once we take them out of the freezer, we have to slowly thaw them so that they don't die. And so this will thaw them slowly to body temperature and keep them warm. We also heat up all our media this way and everything else so that the cells are cozy in a little bath of nice warm media instead of being shocked into cold media when they go into their flasks. So this is one of our oncology patients today, and she has multiple tumors here. As you can see, every one of these little red bumps is part of the same tumor that's in her skin. This is a mast cell tumor, which is the most common skin tumor in the dog. And this has also spread to her lymph node and even to her spleen. At this point, I don't have a current clinical trial for this type of disease. However, clinical trials in the past have focused on specific mutations in growth factor receptors for this type of tumor and how to inhibit them. And there are new drugs that will be available on the market soon that may help dogs like this out with these types of tumors.
Heather, I understand you also do preclinical trials on human cancer, in treatment potential for human cancer. And you do this in conjunction with MD Anderson Hospital, Cancer Hospital in Houston. Is that right? Can you tell us a little bit about what you do there?
Yes, sir.
Okay.
So we have the second clinical trial we talked about with lymphoma is actually a clinical trial that's being sponsored, and the work is being done here and also with the lab at MD Anderson. And the thought is that because dogs and humans both get lymphoma, if we can make this work in the dog, we can probably make it work in the human as well. Dog diseases progress a lot faster than human disease, and so getting those kinds of results are good while still being beneficial to the dog, but we can get it in a timely fashion. The other thing that's important about our dog model that we can offer that other places cannot is that tumors in our animals are occurring spontaneously. And so these animals are getting cancer for the same reason that people get cancer. And for that reason, they're actually a real test of the types of drugs and therapies that we want to test. When you take an immunosuppressed mouse or a mouse that doesn't have a good immune system and you give it a human tumor, that's a false environment that you've created to test your theory. And it doesn't always work the same in the lab as it does in the clinics, whereas our dogs are a better predictor of that. And so that's why we use this particular model for this kind of preclinical therapies. The good thing about those, they can definitely inform human clinical trials, so that when we test these theories in our dogs, what works for them and doesn't work for them can be immediately applied to human clinical trials to make them go faster and to have fewer side effects and to get the scheduling right quicker and things like that. We have some good ties with them. We actually have a meeting coming up this week with 15 different scientists from MD Anderson who are coming down here to work on collaborations. We go there all the time and meet with them to talk about possible collaborations. We even have meetings or conferences that we set up a couple of times a year where everybody presents their data and people get together in working groups to say who can help who with what, and write grants together and things like that. But we are uniquely placed here as the only vet school in the state of Texas to actually provide that kind of clinical model.
Okay, thank you. Well, that's very exciting work you do, and I'm sure a lot of good is going to come from it.
We hope so.
Thanks again.
Thank you.
As I said, in preclinical trial studies, the investigations are usually done in experimental animals. And these animals can include things like fish, but usually it's mice and rats. The purpose of these preclinical studies is to, first of all, establish that the results you expect actually occur in a whole animal. And the second purpose is to get some idea as to what the range of doses are. And finally, to establish what's the highest dose that's still safe and determine which doses are dangerous to administer. Once this information is at hand and your criteria are satisfied, studies in humans can begin, and these are divided into 4 phases. The first is to test a small group of people for the first time to evaluate the safety of the drug and to determine the range of doses that are safe to use, and to identify any obvious side effects. All drugs, by the way, have side effects, and you need to know what these are and how serious they are. Phase 2 is a test in a larger group of people to verify that the drug is effective and to further evaluate its safety. Then in phase 3, you have very large groups of people involving thousands to confirm that the drug is effective and safe, and further to have a large population in which to determine side effects. At this point, the drug can be marketed, but even after marketing, there is a phase 4 where the drug is studied to identify risk and benefits and other ways of using the drug in the general population.
I'd like to introduce you all to Dr. Tim Phillips, who has just won a national award for his research involving clinical trials on treatment for a toxin that is rather common.
In our lab, we work with compounds called mycotoxins. They're produced by fungi and molds, and they're very important in that they have been shown to cause disease and even death in man and animals. And probably the most significant, and maybe the most studied of these mycotoxins are known as aflatoxins.
So I guess, Tim, that's my first question I'd like to ask you is, this toxin you've been studying for many years?
About 20 years.
About 20 years. It's called aflatoxin. Can you tell us a little bit about what that is and why you've been studying?
Yeah, aflatoxin is one of probably, well, really more than 300 mold-elaborated toxins that can cause disease and death in humans and animals. Aflatoxin is probably the most studied and maybe the most significant because it's one of the most potent liver carcinogens known to man. And it's a problem also because it occurs after drought. So those areas, those countries that are susceptible to a lot of drought stress in their corn and peanuts may see more of this compound ending up as a contaminant of animal feeds and human foods.
Now, why does it occur after drought? I thought molds grow better when it's wet.
Yeah, that's a really good question. And actually, there's probably a lot of ways to answer that, but in general, aflatoxin, the fungus, likes to produce the toxin at about a maximum of 18 degrees. And
centigrade.
Yes, and when you're out in the field, like in corn, for instance, normally, at the dough stage or whatever, before you bring it in, you'll have high moisture content. But during drought, that tends to drop the moisture drastically in the corn down to the area that the mold really likes. And of course, what happens when they bring the corn and peanuts in, the way they try to stop it from being a storage problem is to dry it down to 13% moisture, to keep it away from 18. But the drought tends to shift it that way more rapidly.
So in the early years of your studies with aflatoxin, I guess you could think of those as preclinical studies, although maybe at the time you weren't thinking of it that way because you didn't realize that the pharmaceutical products would come out of this. What kind of preclinical research did you do?
Well, the type of work we've done with the aflatoxin has been bench work. A lot of what we call in vitro mechanistic studies to determine the mechanism of how this clay works to bind aflatoxin in the stomach and intestine. And we've looked at a variety of animals, especially with our colleagues at USDA, poultry and swine and lambs and dairy cows and caprine, dairy caprine.
Why did you use so many different species?
Well, because there's a big variation in how these are affected by aflatoxin, and in order to know you're dealing with a compound that's safe as you transition this into humans, you need to have a lot of species. We even use modeling procedures to model the toxin and model the clay and learn more about it, and even looked at thermodynamics. Now, what we did learn was that these were very selective based on our thermodynamic work, and that
What, clays?
the clay, this particular clay we call Novacil.
How did
how did clays get involved here? What caused you to start working with clays?
Well,
In context?
we were looking at a lot of different sorbents, trying to find something, including antibodies, even a biological system that would bind this and activate it in the stomach and gastrointestinal tract. So the target was to develop an enterosorbent. But antibodies didn't work well, and other sorbents didn't work, and we found this clay.
These are some clays collected in Ghana, and these are edible clays that people used to eat in Ghana. And you can also find these pieces of clay. This is more like in a natural source. You find them with this shape, and the clays that we used for the study were placed in capsules like this. So people didn't know if they were taking the placebo or the clay treatment.
But a lot of clays are bad for you.
That's right. You have to be extremely careful because they come out of the ground, and just like plants and soil, you know, can have a variety of priority metals that can be carcinogenic, arsenic, you know, cadmium, mercury, lead, and a variety of others, as well as dioxins and furans.
All of that's bad stuff.
All that's bad stuff, and so you have to, our particular clays that we looked at, we very carefully characterized for that, and we even have a process now where they're processed and they're refined to be sure that the levels of these toxins are less than what you would get in food. They're checked in animals for safety, and then they're checked to be sure that they don't interfere with nutrients, and Novosil doesn't. So we're very, very happy, you know, that it binds aflatoxin very selectively.
Now, Novosil, apparently the name you've got for this pharmaceutical.
Correct.
It's in clinical trials now.
Correct.
What's
Correct.
the status of your clinical trials?
Well, the clinical trials that we're doing now, well, the first trial we did, in order to go to Africa because of the sensitivity when you're doing clinical trials, we ran the first study in the U.S. in Texas, and it was a phase 1 trial, which we call an adverse events trial, and we were looking at, you know, up to a high dose of 3 grams of this clay a day in a person, and to determine were there any adverse
Taken by
effects.
mouth, right?
Taken by mouth, capsule by mouth. To determine were there any adverse effects, you know, based on health science center doctors, you know, measuring clinical measures and blood measures, and urine measures, and we found it was very efficacious and very safe. And that was the first study, and then recently we finished what we call a phase 2A study, and that was a 3-month intervention in Ghana. This is Ghana, and it's bordered by Togo, Upper Volta, and the Ivory Coast. We fly into Accra, the capital city down here on the coast, and then we drive for about 9 hours over really bad roads up to the northern Ashanti region, which is where our site is for this second study. And what our results were in these people at the end of this study was that when we looked at biomarkers in the blood and urine, and those biomarkers are metabolites and adducts that we know give us an idea of how bioavailable aflatoxin is, or how much that person was exposed. When we looked at those, the biomarkers in urine for aflatoxin exposure and in blood were significantly decreased. And it was based on those people taking capsules that had just enough Novosil to just cause an effect, a minimal effective dose, just to be sure we were safe. We only used a minimal effective dose that we had seen that just barely caused an effect in animals. And that's what we used, and there was greater than a 40% decrease in exposure in the people in this trial.
Now, were the people actually deliberately given aflatoxin, or were we more or less treating them for the aflatoxin that they normally?
Yeah, just treating them for the aflatoxin they're normally exposed to. When we selected them, we looked at about 500 people, and the first thing we did was the biomarkers of exposure, the bloods and urines, and we saw that everybody in this population had biomarkers in their blood, and they eat. Probably the reason for that is they eat a lot of peanuts and corn. These types of commodities can generally have higher levels, especially if they're moldy. And you know, it seems that this toxin impacts the poorest people more so because they probably end up having to eat
So that explains why you went to Africa, because they have to eat this food. It happens to
Exactly.
be both.
Exactly, and so they're what I would call a population at high risk for aflatoxicosis and liver cancer. And so there's a great need there, to be able to come up with something that is sustainable and environmentally benign, and even in this case, culturally acceptable. That was really a neat thing because, again, they eat clays. These clays aren't the same as Novacil, but they're used to eating clays. So clay ingestion or geophagy is culturally acceptable in these countries on the continent of Africa. And so I think that gave us also an edge in being able to have them accept this technology.
Now back to the earlier point. The humans are getting exposed to aflatoxins normally.
Right.
As part of the daily existence.
Correct.
When you did the animal studies, you had to give aflatoxin to the animals, right? To test the effectiveness and safety
You bet.
of the clay.
You bet. Yeah, to be able to do that, because aflatoxins, if you buy them when they're synthetically produced, are extremely expensive. So we, with our colleagues at USDA, we inoculated the fungus that produces, an aspergillus that produces aflatoxins, in a rice powder. And after growing it out for a couple of weeks, we produced very high levels in the rice powder of the naturally occurring aflatoxins. And then we mixed that in to their diets to produce the effect we saw, and to be able to prevent it with the Novacil.
Now, Novacil is getting close to being a commercial product available in the marketplace.
Well, we're certainly hoping so. It still has some more stages to go through. We were just refunded for another 5 years in Africa to do a more extended study in humans, a 1-year study, and a preliminary study that I'll be starting in a couple of months in Ghana is really of interest to me. And it's a pilot study before the 1-year study, and I'm going to be able to show if the clay, when put in a powder form in peanut meal and cornmeal, and then when it's used to cook the food in the villages, if eating that food with either placebo or the Novacil, makes a difference in the exposure. Because what we've done so far is give them a capsule before each meal. Now I want to give it to them in the meal. Make sure that the processing to make fufu and banku and the dishing, kadia, peanut soup and stuff like that, if that won't affect the efficacy of this. And if that's the case, then we'll be able to apply it in the food. I think that'll be probably the best way down the line.
Well, it seems clear to me that Novacil should help prevent the hazards. It occurs to me that animals eat food that is contaminated with aflatoxins.
Right.
Pigs eat
Right.
moldy corn and stuff
Yes.
like that. Are there implications here for animal treatment?
Oh, most definitely. Recently, well, I guess it was a couple of years ago, and then maybe 6 or 7 years before that, we had outbreaks of disease even in domestic animals, in dogs. I mean, you may have heard that. I think the last one was in the Carolinas, and it was due to corn that had been drought-stricken, in the field, and loaded up with aflatoxin, and dogs started dying. And I get calls all the time with animals, that are having problems with this, or presenting at the vet school with livers that are classical, for aflatoxicosis. So I would love to see this be able to go into animal feed and be able to protect dogs and as well as farm animals.
Is it going to be dirt cheap? Tim, that's wonderful work. Thanks a lot for sharing this information.
Thanks, Bill.
So I think you can see from the 2 lab visits that we just had that research on animals leads to treatment for animals, and very often it's done with the idea of developing treatments for humans. You've also seen that research on humans can lead to treatments that can be used with animals. So in other words, it's all one medicine, after all. I hope you've enjoyed this tour, and we'll give a lot of thought and appreciation for what's involved in developing new medicines and treatments.